Now, our next player in the translation process is the tRNA, and Francis Crick very aptly named these adapters because in all essence, that's what they do. Essentially, they look at the mRNA and they adapt it to the correct amino acid. So it's like they can plug into a codon on one end and on the other hand have an amino acid. So we recognize then that the role that they play is very much in recognition. And if we were to take a tRNA and literally flatten it out on a page, get rid of all of its three-dimensional structure, we would see something called the clover leaf structure, and that's what you see on this page. There are a couple of important regions within the clover leaf structure. You can see over on the right is the D arm, on the left is the Tcc arm, and the Tcc arm is known for having some really unusual nucleotides, ribothiamidine and pseudouridine. They're just out of the ordinary. There's also what we call a variable arm, and the variable arm, yes, does vary in length depending upon the tRNA. And then the most important arm, arguably, or maybe second most important, depending on how you look at it, is the Anticodon arm. The Anticodon arm, of course, is what does plug into the codon, so to speak. It's what recognizes the codon on the mRNA. So that being said, we could actually look at this anticodon and realize that it runs antiparallel to the codon that it will recognize. So that being said, we can quickly jot down the termini on this particular anticodon because we know that the codon has a 5' end over here, right, and the 3' end over here. So the anticodon must be antiparallel to that. That is, this must be the 3' side, and this must be the 5' side. Now, if we follow that all the way through and the whole tRNA, we would recognize this to be the 5' terminus and this to be the 3' terminus, and it turns out that the 3' end is where the amino acid attaches onto the tRNA. So now that we've recognized that, we can go ahead and write down what the codon would be based on base pairing rules, right? So U is going to pair with A, and then finally C is going to pair with G. So we recognize that the codon that tRNA would bind with would be AAG. So in order to determine what this amino acid is up here, we simply look up AAG in our codon usage table. So let me get rid of some of my chicken scratch here, and we'll write that down. So that's going to be AAG, and we'll use our codon usage table really quickly, see how we're doing with that, to look up what amino acid will be attached to the acceptor stem at the 3' end. So we'll look up A in the first position, A in the second position, and then finally G in the third position. So recognize then that the amino acid that would be carried by this tRNA would be lysine. So what's kind of cool actually is that even before a tRNA comes to do its job as an adapter, the first thing that actually has to happen to a tRNA is to get its amino acid attached to its 3' end. So we call that activation of the amino acid. So the amino acid gets activated by attachment to the appropriate tRNA. And this is, Eric's going to appreciate this because it's got sort of this medieval note to it, where essentially a tRNA is named based on the amino acid that it's destined to carry. And then once it does carry that amino acid, we indicate that in the name too. So upon activation, we say that the amino acid was, or that the tRNA was destined to carry lysine. And then once it carries lysine, we say that. So we call this lysyl tRNA lyse. Before it carried the amino acid, we say it was tRNA lyse, destined to then carry lysine. And when it does, we say that it is carrying that amino acid. So it's just got an interesting sort of note to it in order to indicate whether or not the tRNA has been bound to its amino acid or not. And there's actually a whole group of enzymes called amino acyl tRNA synthetases that add the amino acids onto the tRNAs that they're supposed to be added onto, which is really interesting because they have some recognition for different regions within the tRNA. So cool stuff, right? So we recognize then the tRNAs working as adapters. Let's practice one more time to see how we're doing with this idea between the relationship of an anticodon and a codon and how it dictates the amino acid carried. So we'll look at another one. What amino acid does a tRNA carry if it has the anticodon sequence 3' GCG? Okay, so wait a minute. Now we have to draw this out in order to feel comfortable with what we're doing, because we want to remember that. Let's draw just the anticodon arm. So here's the anticodon arm of this tRNA. And we remember that if we kept on drawing, up here would be the 3' end and then the 5' end. So we need to put the most 3' end of the codon in this region. So we would have G, C, G, so that then it could recognize the 5' side of the codon. So then the codon would be C, G, C, right? So anti-parallel relationship, don't forget to do that to make sure that you're pairing the anticodon and the codon correctly. And then remember that what we look up on the codon usage table is always the codon. You don't look up the anticodon. So now we'd want to pull up our codon usage table again and look up CGC and see what that amino acid would be. So C, first position, G, second position, C, third position, CGC, arginine would be the amino acid that was carried by a tRNA with that anticodon sequence. So practice that a lot so that you understand just how tRNAs can act as adapters. This is a picture of the actual three-dimensional structure of a tRNA. So in order to see that clover leaf structure, right, this would have been what was flattened out onto the page. And in fact, in actuality, the tRNA carries a lot of tertiary structure. So it winds around and you can see local regions where the tRNA hydrogen bonds, and it forms even a bit of a helical structure in regions throughout. So this is more what the overarching shape of a tRNA would look like, more like an L or a liver-shaped sort of structure that you see there. So one note is that tRNAs are partially responsible for degeneracy. Remember degeneracy, the fact that there's more than one codon that dictates a particular amino acid. And that's because although there are 64 codons, there are not 64 tRNAs. There's 40 some odd tRNAs, which must mean that sometimes a tRNA can recognize more than one codon. And it does that because it gets kind of loosey-goosey in its most 5' position. That would be, of course, the most 3' position of the codon. And if we remember back to our codon usage table, remember that that's where the flexibility took place, because always those first two nucleotides would stay conserved in degeneracy, but then the third one would be the one that would change. So this actually makes quite a lot of sense. So when we recognize the root of degeneracy here, we can say a couple of things. One is that there's no tRNA for a stop codon, but then another is that sometimes tRNAs will recognize more than one codon. And here's a bit of an example. If we look at the anticodon 3' CCG, we would recognize that one of the codons that would recognize would of course be 5' GGC. So we would expect it to recognize 5' GGC. But on the other hand, it turns out that right here in this position where the G and the C hydrogen bond, there's actually some flexibility right there. That G can get loosey-goosey actually and recognize more than one nucleotide right in that particular spot. So this is something it's a phenomenon called Wobble. So let's write down, first looking at our codon usage table to see what other codon besides GGC might be recognized here. So looking for GGC in the codon usage table and then recognizing G, G, see glycine, right? But recognizing that perhaps that tRNA also recognizes GGU because it also encodes for glycine. So in fact, this tRNA does recognize both of those. And this is a phenomenon called Wobble. I love that. Wobble. This is your next smooth line on your next date can be, you just have nothing else to say and you say, Wobble leads to degeneracy. And I mean, that person will be all yours forever. All right, now that we understand a little bit more about the root of degeneracy, let's move into the process the three-step process of translation. Now, remember that in both replication and transcription, we had those 3 steps, and that's going to be no different in translation. So those 3 steps, remember, were initiation, elongation, and termination. So we're going to go through all of these a little bit at a time, beginning with initiation. So we'll be looking at the process by which the whole process of translation gets started. Let's begin by making a quick note of the difference between prokaryotes and eukaryotes, in that in prokaryotes, the process of initiation of translation can begin before transcription stops. So this is shown on this cartoon drawing. It's obviously not to scale, but what we see is our RNA polymerase enzyme shown in orange there running down the DNA and reading the DNA, making the mRNA transcript, and even before that RNA polymerase has fallen off and terminated transcription, the ribosome has bound to and begun translation. So it's can be sort of a simultaneous process. And the polypeptide chain begins to be synthesized, even prior to finishing the mRNA. Now, this isn't possible in eukaryotic cells, so think about that for a minute. The reason that this isn't possible in eukaryotes is because of compartmentation. So if this is our eukaryotic cell over here, and we remember that it has a nucleus, and of course that nucleus with its many nuclear pores, this is the site of transcription inside of the nucleus. So transcription takes place in there. Whoops. And then in contrast, after the mRNA is made, then that mRNA can be popped out of the nucleus, right? It can be sent out, and then you get the mRNA out here where it can be translated. So because of that, transcription and translation cannot be simultaneous the way that they are in a prokaryotic cell. So things are a little different. But let's begin then by talking about initiation of translation. That is, where does this all start? And we know one thing that's important is that it starts at the AUG start codon. But something has to happen to orient the ribosome and all of the translation machinery at the AUG start codon. So one of the biggest things playing a role in that is going to be the 30S ribosomal subunit, and it's actually going to recognize a region on the mRNA that's given a special name. It's called the Shine-Dalgarno sequence. Now, obviously that's just named after the discoverers, but what it really is this region that I've shown in sort of a teal blue right there, that Shine-Dalgarno sequence generally tends to be a purine-rich sequence. So a purine-rich region of nucleotides. So AGGA or something like that. And within the 30S subunit, here's the major function for the 16S ribosomal RNA. It has a complementary sequence on that RNA that recognizes right there and kind of holds on to, latches on to the Shine-Dalgarno sequence. So that sequence, of course, is going to be a pyrimidine-rich region. Oops, I spelled purine wrong here. Let's fill that in. Purine-rich region of nucleotides. Okay, so the pyrimidine-rich region in the 16S ribosomal RNA latches on and holds on to that, and it's what allows for the sort of beginning of translation to take place, the sort of culmination of a lot of events at that one site. So it also does orient such that the AUG start codon, which I'm showing in bright pink here, is going to be in the right place to initiate the beginning of the reading frame of translation. So we'll go ahead and note some of the other things involved with this. Of course, we know that the tRNA has to recognize that AUG start codon, and we also know that it will be carrying F-met, or the first initiator amino acid, N-formyl methionine. So recognizing that at the AUG start codon, this is where the initiation complex is coming together, and the N-formyl methionine, or F-met residue, of course, is what's the amino acid that's carried by that tRNA that's binding at that start codon. Now, these are not the two only things involved with this. There are actually more. There's more to the initiation complex in the way of initiation factors, and these initiation factors have some pretty interesting roles. For example, IF2, and I'm actually going to jot this down so we can have some idea of what these do. IF2 is going to be involved in ensuring that the right initiating tRNA binds at the AUG start codon. So let's just say ensures binding. I'm writing like a 2-year-old tonight. So ensures binding of correct initiator tRNA. That is to say that N-formyl methionine is the first amino acid to be bound. They're bound to its tRNA. So we recognize that there's a couple other initiation factors as well. IF3, this stops premature association of the 50S and 30S ribosomal subunits. So we know that they're apart until they begin their process of translation. So it stops premature association of the 30S and 50S subunits, right? They really want to be together, but this is the wingman ensuring that loving association takes place at the right time and no sooner. Your IF2 or your IF1, this one actually kind of coordinates the functions of some of the others. I'm going to put it as a coordinator. Coordinator. And it is thought to play a bit of a role in the leaving of initiation factor 2 and the hydrolysis of GTP in that initiation factor loss. That's a probably really important thing to note right there, right? Is that with each event in translation, it's very endergonic, right? It's very energy consuming. So right off the bat here, we see a GTP consumed in just the initiation process. So translation is one of the major processes that is powered by all of that ATP and GTP maybe that was synthesized within all of those catabolic pathways that we spent so much time talking about. Okay, so now that we have the initiation complex all ready to go, clearly the one thing that's not happening here is that the 50S subunit hasn't associated yet. So at this point, GTP will be hydrolyzed as IF2 leaves, and the 50S ribosomal subunit will come in and be associated now with the 30S subunit. So we'll see that in the next phase, where the 50S ribosomal subunit joins as the initiation factors leave. And now we have synthesized, or we have formed, what is a full ribosome ready to head on down the mRNA, reading one codon after another and beginning to build that long polypeptide chain. So at this point, we go from initiation into elongation, and that'll be our next step of the process. Now, before we really get rolling on elongation, I want to see if we're going to make good ribosomes. So see if we can figure out how to read an mRNA and determine what the resulting polypeptide chain will be. So here's a practice question where we see this mRNA down here, and rather than writing out the Shine-Delgarno sequence, I've made it look like a black box there, but we know that that black box is actually a purine-rich sequence. Maybe, for example, it is something like AGGA that is recognized by the pyrimidine-rich region within the 16S ribosomal RNA of that 30S subunit. So we know that that's where the 30S subunit will associate and begin that process of all of the translation machinery coming together for initiation. So let's read this question and make sure we're understanding what we're wanting to do. So the only time AUG serves as a start codon is when it is downstream from a ribosome binding site. That is the Shine-Delgarno sequence, which is right there. So the first time that we see that AUG start codon after the Shine-Delgarno, we know it will serve to set the reading frame and start translation. Otherwise, it just encodes for methionine. So a ribosome binds to the following mRNA molecule at the site indicated by the dark box. Determine where translation will start and what are the first 3 amino acids that will be incorporated into the polypeptide. Easy enough, right? As long as we remember that translation always begins at the AUG start codon. So we want to make sure that we go and find the first AUG downstream of the Shine-Delgarno sequence. So we'll scan down and be looking for that first AUG. And as we scan, here's the first A. Oh, there it is. The first AUG. So that will set the reading frame. So from here now we can start to write the sequence of the polypeptide chain, knowing that it always starts with the amino terminus first. Of course, in prokaryotes, that amino terminus is formulated. It's going to be N-formal methionine, right? But we can say that that first amino acid will be F-met. We also know that that AUG start codon set the reading frame. So the next amino acid will be GUC. Okay, go ahead and look that up in your codon usage table. I feel like I should have elevator music or something while you're looking that up. Go ahead and look it up. GUC. And I think that it turns out that our next amino acid should be valine. And then look at what happens now. AUG again. So the second time it's encountered, it just encodes for methionine. So just a plain old methionine, not modified in any way. So we could keep on going, right? And I suggest that you do the GGA, the AAC, and keep reading in triplicates and finding out what amino acids are going to be next as we head towards the ending carboxyl terminus. Now, if you guys really want to rock my world, I hope you will. Please remember that proteins have amino and carboxyl termini. They do not have 5' and 3' ends, right? Only nucleic acids have 5' and 3' ends. They have a 5' phosphate and a 3' hydroxyl. But proteins don't have that. Remember, they have an amino terminus and a carboxyl terminus. Now, to help you remember that, I've gone ahead and made a cheesy drawing. So here's the actual molecular depiction of what we just wrote out. So here's our methionine residue, right? You recognize that R group on methionine. And then we recognize that it's formulated. So this is the formal group that's been added onto the methionine. And then our next amino acid, C, is valine. And I should have probably circled that as part of methionine, right? It's carboxyl group. There's valine, so there's methionine. And then we know it's formulated methionine, so it's F-met. Then there's valine, that nice hydrophobic amino acid, right? And then we have another methionine. So please remember when you write out amino acids and their termini that we always, in any polypeptide chain, we always have an amino terminus, then we have the sequence, and then a carboxyl terminus. A lot of times students make a mistake in putting a 5' and a 3' end on proteins, and proteins don't have 5' and 3' ends. So please keep me sane and don't put 5' and 3' ends on your proteins. Okay, so have a fantastic Monday, and I can't wait to see all of you tonight at office hours and again on Wednesday for our next vodcast.